Okay, Morgan, it is late, so bear with me, but I'm going to walk you through your entire methodology for your project. This will be a long video, so take it step by step, pause it anytime you like. But this is your project from start to finish, including how to mix the chemicals, how to prepare the media, how to treat the worms, how to treat your bacteria food, what exactly the bacteria food's going to do. It's all boiling down to the fact that we are feeding your worms a bacteria that have a plasmid in them while also knocking out that plasmid as much as possible to see if there's a phenotypic change with your hydrocortisone treatment added. So, here's the purpose of your project. You are going to test whether or not hydrocortisone, which is the cortisol analog that we can buy, affects the success of gene silencing in C. elegans. Gene silencing is one version of gene therapy we can do. An easy way to think of this is if I had a sickle cell anemia gene that I wanted to uh, prevent from being produced over and over again as a protein, then I can silence that one gene by just keeping it from being expressed. And that's a type of gene therapy that we can do. Some facts that we can use to kind of understand exactly what's going on. We're going to feed the C. elegans with a bacteria strain of E. coli called HT115DE. The numbers and labels is not really important, but what is important is what they do. They produce double-stranded RNA that targets a specific gene to deactivate it. That gene, funnily enough, is called UNC22. So when UNC22 is silenced, that worm that is getting this gene is going to develop this twitching motion that we can count to see how much of that gene is expressed. But when it's silenced, or when it's activated, the worms won't have a twitching phenotype. They'll have a normal phenotype of doing nothing. So by exposing the worms to different hydrocortisone concentrations, we can measure whether the stress hormones increase or decrease or have no effect on the gene silencing efficiency, aka the amount of twitching that's occurring as a phenotype when this UNC22 is silenced. I do have a video that I can show you about RNAi, which is RNA interference. That's what is happening here with the bacteria and the double-stranded RNA with the UNC22 gene. And I also have a video showing you exactly what that twitching is going to look like after an RNA interference against UNC22 is established. The list of materials is actually pretty simple. It doesn't look simple, but what we need is C. elegans, of course. We're going to get a wild type strain that we call N2. That's going to be from University of Minnesota. They have a specialized group that makes sure that C. elegans can be used all across the world. That's called the Canarhydritis Genetics Center. We can also get the E. coli strain, the HT115DE, from the CGC, and we're going to get two versions of it. One is going to have that UNC22 gene that is going to give us that twitching phenotype. The other is going to have a plasmid in it that doesn't have the UNC22 but has everything else that the plasmid is supposed to have, such as antibiotic resistance and a promoter gene and stuff, which we'll use as a control variable. We might not be able to get this one. I don't know if the CGC has that or not. If not, we just won't have a negative control. We'll just have a baseline of 0 and 25 and 50 and 75 and 100 for our hydrocortisone amounts. We will also need to make NGM agar plates. You'll get bottles of NGM, that's nematode growth medium, NGM for short, and you'll pour those into agar plates. We'll also need to make some LB broth and LB agar so you can grow these E. coli strains. And we'll add in some ampicillin and tetracycline because this bacteria is resistant to both of those um, antibiotics, so we can specifically make sure that our bacteria plates do not get cross contamination or any other bacteria strains in it. We can select for these individual E. coli with these two antibiotics. Growing them in that medium with those antibiotics will help us determine, yes, this E. coli strain is the one with the UNC22 and the L440 plasmid. IPTG, this chemical, is a chemical promoter, which you may have done the MI GFP lab last year when you were in MI at your other school, where you transformed bacteria to grow glow in the dark. IPTG is a chemical promoter we used in that lab to have the bacteria make that protein that makes them glow in the dark as much as possible. Here, we are having them make the product for the UNC22 as much as possible so that there's as much twitching phenotype as possible. We'll, of course, have to get hydrocortisone powder. We'll dissolve it in ethanol and make a stock solution for you. We'll have a stereo microscope that you can use, easy enough. We'll have plenty of ethanol for cleaning, and we'll have plenty of pipettes, sterile tips, inoculation loops, labeling materials, all that dust stuff. We will have it. There's some other things on this list that are or that are not on this list that you'll actually use, such as something called an M9 buffer, but I'm pretty sure we have everything that would not be on this list. So here's the experimental design that we came up with when we talked about percentages the other day. The other day we talked about, hey, if we figure out a dosage amount at 100%, then we could scale down the different groups of hydrocortisone by 75%, 50%, 25%, and 100%. Well, I figured it would be easiest to go with a hydrocortisone amount that wouldn't kill um, the C. elegans or the E. coli that they're eating right away. And I did some research and came up with a concentration of 100 micromolar as the final concentration of hydrocortisone that you're treating your plates with, which is actually kind of nice because then you can just say 100 micromolar, 75 micromolar, 50 micromolar, and 25 micromolar are your treatment groups here, and 0 is going to be your baseline. We'll also have another 0 hydrocortisone to see how the negative control reacts if we get that one with the empty plasmid without the UNC22 gene. And remember, we are trying to find the percentage of worms that are showing the twitch phenotype. So based on this, the ones that have the most successful UNC22 being established in them would be the ones that have the most twitching phenotype. And that could either be ones that were exposed to no hydrocortisone or lots of hydrocortisone, or it could be equal in all of them. We don't know yet. I do think you should run replicates of this so you can either average out your results by the end of this or at least have a fallback plan in case one of your plates do not work out the way you expected. So that would be 3 plates per edition. If we ran it like this, that's 6 plates, 3 plates per each one, that's 18 plates. And you would need extra plates for subculturing the worms along the way, as well as subculturing the bacteria along the way, but this is not that many plates at all, and you're only going to have 50 worms about to count each time, so it should be a relatively simple lab, even though it seems very daunting right now. You'll be like, oh, yeah, I can do this. So, let's talk about each part as you prep for the lab and get ready to do the actual worm experiment itself. The first part is that we need to plate some nematode growth media. We need to prep the plates that the worms will live on. We will get you bottles of this nematode growth media pre-made that all you have to do is you set up a bunch of plates, we'll say 18 plates or so, onto a tabletop. You should first sanitize the area and make it as sterile as possible with ethanol and keep those plates closed because as soon as you open them, they become unsterile or can become unsterile. Then in that bottle, it's going to look like jello set up on the inside. So we're going to unscrew the cap until it's just about ready to screw off and lift off the top and just kind of stick it on top while it's resting there. Not screw it back on top, but like screw it off and just like fiddle with it a little bit to make sure it doesn't fall off, but so that it is on top of the bottle. So that cap is going to be resting on top of the bottle spout. The reason for this is we're going to melt this agar, this nematode growth media, down in the microwave to liquid status. And if we have that screwed on all the way, then the air that's inside of that bottle will actually end up bursting and it will break the bottle and break the microwave, and nobody will be happy about that. But also, we need to make sure that we pay attention to this as it's microwaving because we want to get it to about 55 degrees Celsius, and we don't really want it to boil over, else it makes a big old mess. In addition to that, it ends up making it less sterile because all the air that's escaping out of the bottle is then replaced with unsterile air going into the bottle. So we're going to have to watch this very closely to make sure it doesn't boil over, but it is indeed melting. So it will boil a little bit, but I don't want those bubbles to reach the top. We want them to stay in the bottle. We'll have to do that with like 15 to 30-second spurts, and you'll have to reach in with a glove, a hot glove, and swirl it around a little bit while keeping the lid on top. Don't worry, I'll be there to help you the first time around. Once it's completely molten, however, you can go ahead and pour that molten agar directly into the plates. We're going to aim to just cover the bottom of the plate, or I can give you my expensive pipette controller so that you can draw up about 20 milliliters of agar for each of the plates. But I should warn you that that pipette controller is super expensive and you will be responsible for buying it if you break it. And it is easy to break. I have had students break it before. After you pour in the molten agar, you just simply keep the plate closed and let it sit to cool and solidify. There should be some condensation on the top, but as long as we don't jostle the plates around, that condensation doesn't drip down, back down onto the plates. We will keep these in the fridge for as long as we need to before these worms get on top of them. And I'd say we will go ahead and make at least, I'm talking maybe 20 plates or so. So you're going to have quite a few bottles to melt down, especially if you do measure out 20 milliliters per plate. That's about 5 or 6 plates per bottle. So if you're doing 20 plates, you got to have at least like 3 or 4 bottles worth. It's a lot. Here is the hydrocortisone plan that you're going to have to make that 100% level. It's really 100 micromolar in chemistry talk. I'm going to have a 10 millimolar. Remember, a milli is 1,000 of a micro, so this is really like saying 10,000 micromolar instead of 100 micromolar. It's like 100 times as big. You're going to take 2 microliters of this stock solution that I'm going to make you and add it to 198 microliters of sterile water, and that will be your 100 micromolar solution. And you'll just keep that in a tiny little bottle, and you'll be able to apply it to your plates each and every time. To make a 75, you just simply make 75% as much hydrocortisone, so 1.5 microliters instead, and 198 with sterile water, and adding in 100% ethanol, that would be 0.5 microliters liters of ethanol. The reason we're adding a little bit of ethanol here, don't worry, this ethanol will not kill your worms and will not kill your bacteria. It's going to be diluted as enough in this whole solution here. But the reason that we're making an addition of ethanol here is because when I make this hydrocortisone stock, it actually has ethanol in it. So we're counteracting the difference in the lowered amount of hydrocortisone with the increased amount of ethanol to make a more dilute solution. 50 micromolar is going to just be 198, and 25 is 0.5, 1.5, and 198. You'll follow this every single time you want to make a plate that you're going to treat with hydrocortisone, because once you've mixed all of those things, you're going to pipette those 200 microliters onto each NGM plate surface. So if we have 3 plates per or per condition, let's say you have 3 100 micromolar plates, then that means you'd have 600 microliters total to pipette onto those 3 plates. Then you're going to tilt the plate around to spread it evenly, just roll it around in your wrist a little bit, and you'll leave the plates cracked open during this time. So we're going to have to do this when there's not a lot of movement in the classroom, or maybe put it in the closet so there's not any chance of contamination for about 30 to 60 minutes to dry. So you could do this during our second hour and then be able to put them away later in the day. We'll store them at 4 degrees Celsius, which is the fridge. Make sure you label them each and every time you do so, because you will not remember which one's which. Label it with concentration and date. That way you know how long your plates have been sitting there in this concentration, and whether or not we need to redo it because the hydrocortisone might be out of date already, expired already. So that's the worm plates. Now, let's talk bacteria plates, because you have to feed your bacteria. And I really think that you probably won't have to do bacteria plates all that often. But let's say you had to make 6 plates of bacteria, as in um, 1 plate 1 week, 1 plate another week, 1 plate another week. If we make the 6 plates, then you can just subculture them when they get old and the plates are already ready for yourself. What I'll have you do is make this from scratch, and you'll have to put a clean 250 ml glass bottle out on a balance and weigh it out, or we can use a weigh boat and weigh it out. You'll have to weigh out 3.8 grams of LB broth powder. That's the nutrients that the bacteria needs to survive. And then the 2.3 grams of agar, that's the thing that solidifies the nutrients that they want to live in. You're going to add both those powders to the same bottle. Add 150 milliliters of distilled or deionized water. I think we'll just get the distilled stuff and maybe sterilize it for you ahead of time. And swirl it to mix it. Loosen the cap just like you did with the NGM bottles. We'll heat it in the microwave until everything is dissolved and the agar is fully melted. It shouldn't have any particulates in it. And then you let the bottle cool to 55 degrees before we add in our antibiotics. Remember, the antibiotics select the bacteria that we want to transfer the UNC22 gene into the C. elegans. So really, this antibiotic is meant to make sure that no other bacteria from the wild can live in this plate. We will add 1.5 milliliters of ampicillin at the concentration that I make you, and 190 microliters of tetracycline at the concentration I make you. Swirl it up gently. Don't make any bubbles. It's going to be cool enough that you wear it with wear some oven gloves with it, but it shouldn't be okay to use at that temperature. And then you set out your 6 petri dishes and pour about 20 milliliters of agar into each plate. Again, you're covering the bottom basically. Label and date it, store in 4 degrees Celsius, that's the fridge, until ready to use. Now, when we need to work with the worms, you're going to get them probably on a plate, and you will have to transfer them onto one of those NGM plates from before. and maintain those cultures as time goes by. They outgrow their home over the course of like 5 days or so, maybe even 7 days. So you have to have this process ready for them to move into a new home and use those nutrients inside that agar. So once you make these plates, yes, you can let them sit by themselves, but once we get these worms, you have to have the plates ready to go each and every time we transfer them. And that's the goal of what we call chunking worms. When you get your plate, you're going to take a scoop of small agar in a square about 5 to 8 millimeters with an inoculation loop. I literally used the circular end of it to measure out how big the square should be by making a cut with the inoculation loop into a square, using inoculation loop for each of the sides. Then what you do is you use the inoculation loop as a little pancake scooper and scoop that block that you just cut out and flip it onto the new plate. What's going to happen is those worms that you accidentally captured on that square will now crawl off that chunk and establish the new culture, the new home on the new plate. And we need to do this every time that you need to expand or rescue a plate from catastrophe. Maybe we have too much bacteria on the plate and it's overrun with bacteria and killing off the worms, or maybe we need to chunk the plate because they've outgrown the plate and we need to transfer them to a new one. This is how we move worms from one plate to another. This is not how we necessarily harvest the worms when we're ready to actually do the testing. When we want to harvest the worms, we're going to do a process called flooding. Basically, we're washing the worms off a plate into a tube so that you can synchronize them, so you can move them to a brand new plate with no additives added, so that you don't have a random little chunk sitting on top. Or if you wanted to count them, which you will end up doing, this is how you do that. We're going to literally bring the flood of about 1 milliliter of what's called M9 buffer. This is just a salt solution that doesn't dry out the worms when we add it. And you're going to pour it onto the plate, literally bringing the flood to them. And then you tilt the plate gently back and forth to wash that surface of all the worms. You're literally trying to puddle up the worms into a corner and then suck up the worms with a pipette into a tube that we call a suspension because they're now suspended in the M9 buffer. This can also just be called washing the worms off the plate, but I think flooding the plate is more fun to say. I imagine Noah's Ark and such. Now, what happens when you need to feed the worms finally? Well, you made some plates of bacteria earlier, but the bacteria can't just be like scooped off and put onto a new plate. It's not like a lunch meal. Instead, you have to put it into a broth and culture the bacteria, and we have to do some extra stuff to the bacteria to make sure they make the proteins that we want the worms to eat. Specifically, first thing we got to do is make a broth. So you made agar before, and the agar solidified. This time we're going to leave um, the agar powder out and just make an LB liquid, which we call broth. To make the LB broth, we're going to mix 1.25 grams of LB broth powder with 50 milliliters of sterile water inside a big old thick tube. And we'll microwave it and swirl it to mix, and it'll be all good just like that. In fact, actually, we'll probably microwave it in a bottle first, the glass bottle, and let it cool down in that glass bottle before we add the antibiotics. In this case, 0.5 milliliters of the ampicillin I prepare for you, and 62.5 microliters of streptomycin. And we'll store that LB broth in a 50 milliliter tube. With these numbers, we'll end up having a little bit more than 50 milliliters. You'll just have a little bit left over in the bottle that you use. And you'll label it as LB ampicillin tetracycline, LB amp tet. This is going to be your LB broth that you grow bacteria in so that it only selects for the bacteria we want and so that they can be healthy and happy in the swimming pool that we make them. In fact, the next step after that is to take this LB broth and put it into two different tubes, one for the uh, test condition with the UNC22 and one for the empty vector. And yes, you can have multiples of those UNC22s so that you can be ready for as many different testing as possible. You're going to add 5 milliliters of that broth you just created to each tube as long as it is still cooled. You don't want to add it in when it's still hot because if it's still hot, you're going to kill your bacteria very quickly. Then will have your bacteria in some subculture colony, and you're going to scoop up one of those single colonies with a inoculation loop and transfer it into the broth and give it a little swishy swish in there so that the bacteria falls off the inoculation loop into the broth and is now swimming in this liquid, disgusting pool. We'll place those tubes into the incubator at 37 degrees Celsius. Maybe, just maybe, we'll be able to fit a shaker in there so that it tosses it back and forth a little bit, agitates it, mixes it for you, if possible. I don't know. I don't have too many incubators that are big enough to hold that shaker, but we'll see what I can do. And we'll let those grow overnight. And literally 12 to 18 hours later, you will have a tube that looks cloudy with bacteria. It will be full of E. coli that either has that UNC22 protein or doesn't have that UNC22 protein for these things to feed on. We also want to set up a induction process to activate that RNA polymerase in the bacterial plasmid. We want to make sure the bacteria produce as much of that UNC22 as possible and make as many copies of that plasmid as possible to make the gene therapy, the gene deletion, not deletion, the gene, what's the word I'm looking for, silencing as potent as possible. Because if you don't induce this complex to make your bacterial plasmid as much as possible, then it won't trigger the RNA interference in the worms, and the worms will just be like tickled and not actually induce changes to make them twitch. Um, that would be like me giving someone a gene therapy that only is introduced into a couple of blood cells and expecting them to be cured of hemophilia from it. It just doesn't happen when you only affect a few cells. We want to affect all cells. So for each overnight culture, we're going to set up a subculture and take that overnight culture with 0.5 milliliters and add fresh LB amp tet to a labeled tube. We'll then add 5 microliters of 1 molar IPTG. Again, IPTG is a chemical promoter that tells the T7 RNA polymerase to make that UNC22 as much as possible over and over again. For this time, once we have this overnight culture with the IPTG, we'll just incubate it at 37 degrees Celsius. If we can, we'll shake it at that same speed for 3 hours only. During this time, the double-stranded RNA is actively produced. So this is the time where the bacteria are like, oh, hey, I'm being told I need to make UNC22 as much as possible. That's what this is right here. They're making that protein, that RNA, excuse me, not protein, RNA, as much as possible so that the worms can be affected as much as possible. After the 3 hours of shaking and incubating, the bacteria will be ready to seed. So we would take that bacteria culture we just made, this 5 milliliter total and 5 microliters with the IPTG, and we would pipette 100 microliters onto each plate that we have prepared with, or that we are preparing to have worms on, and we would spread that out with a loop. Now remember, you'll have different concentrations of that that hydrocortisol to go into the gels themselves. This is just setting up the gene therapy. So this one tube will cover, looks like 50 plates. You only have to do this really once. But if you don't do 50 plates all at the same time, you might have to do it over so that you can do more plates later on, stuff like that. So after pipetting it on, you spread it with a loop and you let it dry for a little bit, just get soaked into the plates. And then these plates actually go back in the fridge because we don't want the bacteria to grow all over the place on top of them without the worms already ready to go. We want the worms to be able to eat the bacteria as they're growing up, not necessarily while the bacteria are already made and matured. Now it's time to prepare a colony of worms that are all exactly the same age. We'll say L1. L1 is a larval stage of these worms so that when they grow up, they will all be the same age and you'll be able to observe them at a very specific time. So, we first go and find 6 to 10 healthy young adult worms using that harvesting technique where we flood the tubes. We'll take 6 to 10 healthy young adult worms from that little micro tube and put them onto a fresh NGM plate with your hydrocortisone. Then you'll let them lay the eggs for about 1 hour or so. After that, we actually have to go ahead and pick the adults off the plate. So we can do a wash in that case, but you'll end up washing the eggs off as well. So you have to get with that microscope and actually go find them and see if you can pick them off with a pair of forceps that we flame sterilize as well. This will be a challenging part of your lab. But once you do so, since you're only using 6 to 10 healthy worms, you can then leave the eggs to hatch overnight, 12 hours about, and the next day you'll have a whole bunch of worms that will be exactly the same age as larvae, and therefore they will grow up the same pretty much from that point forward. This is so that your results are messy, so you're not observing worms that are at different life stages when you finally do add the hydrocortisone and the gene therapy techniques to it. But we want as many of them to be the same as possible. Then we add the worms that you just made for L1s onto those RNAi plates, the ones you prepared with the bacterial lawn that you put into the fridge. We're now going to add the worms to those plates so that you have the worms feeding on the RNA in their nice new homes. So we can flood the scene again with a drop of M9 buffer and transfer about 50 of those L1 worms to each of the RNA plates. They'll have different amounts of hydrocortisone, but they'll have the same amounts of NGM and RNA. You don't really want to gouge the agar, which is why I would advise against using a sterile pick for this. We could use an inoculation loop, but you probably would not have very much success doing that. So I would say the M9 buffer and flooding it just for a second would be the best choice of possible. You also don't want to cross-contaminate between plates. So once you use one drop of M9 buffer and put it into a tube, you can then just take a different micropipette each time and put them in. Just don't transfer them from one plate to another one. You'll seal the edges with parafilm. If you don't know how to use parafilm, I will show you, don't worry. And we'll incubate these plates inverted for about 48 to 72 hours. During that time, your worms will grow up. And we'll keep them at 20 degrees Celsius. That's their preferred temperature. So here's what's happening while they're growing. The conditions that we need to keep are 20 degrees Celsius for 48 to 72 hours. Once you pick the time frame, please keep it consistent. So if you make 3 plates of 100 micromolar hydrocortisone and you do those first and you do it for 48 hours of growth, make sure the other trials that you do are also for 48 hours of growth. Again, you want the plates inverted. We want to keep them in the incubator to avoid light because hydrocortisone and tetracycline are light sensitive. They will degrade in light. and then you won't have results in your lab. Um, any other time we're using these things, we can cover them up with aluminum foil to make sure that no light gets in. We can even cover the plates up with aluminum foil to make sure no light gets in. We also don't really want to make the lawns too wet or too dry, so we'll pretty much keep them closed the entire time. And keeping them inverted prevents condensation from eking onto the plate and making a big old pool of water there. What's happening during those 2 or 3 days? The worms are eating the bacteria. The UNC22 is going to knock down gene expression, which means that worms will begin showing the twitching phenotype whenever they grow into adults. And whatever your hydrocortisone is doing, it's doing it to interfere with the RNAi efficiency, meaning it will decrease or increase the amount of twitching that we see in the adults. So 48 to 72 hours later, how do you observe the twitching going on? All you gotta do is take that plate um, out of the incubator and place it under the stereo microscope, the dissecting microscope. We'll keep the lid on, but you should be able to see everything that you need to see with the lid on still. Tap it over to the side to agitate them a little bit to wake them up, and you should see some that are twitchers where the worms are shaking rapidly in place, and some that are normal where they're just having smooth, wavy-like movements where they're just kind of snaking back and forth. A twitcher is going to be short-range motion, whereas a normal worm is going to be like moving across the screen of the microscope, forward and back, forward and back. Twitchers are spasming, really. Now, things you want to record during this time, and I can get you one of those counter things so you can record numbers as easily as possible, is you want to give yourself a plate ID and record the hydrocortisone concentration for that plate as well as the bacterial strain, whether it had the UNC22 or it had the empty vector. You'll want to count the total number of worms on that plate, and then you count how many of them are twitching. We can assume that the rest of them are normal, but you still should probably count to confirm the normal one. And we will end up counting a variable called twitch percentage, as in this is the percent of how many of your organisms are twitchers versus not twitchers. And obviously, the number of twitchers will result in a higher efficiency of the gene silencing that we're doing. You can also have some recorded observations because observational science is appropriate, and maybe you don't get numbers that you expect, but you have observations that are different. Who knows? The math that's involved is pretty simple here. For the twitch percentage, all you gotta do is take the count of the number of twitchers that you have and divide it by the count of the total number of worms you have. So it's like an efficiency score. This is how many got twitcher, and this is how many total you have. Multiply by 100, that's a percent. Easy as pie. And we can average your 3 trials for an easy comparison between the 3. That would still be sound scientifically. However, if you want to go win a scholarship, I highly advise you to look into some statistical testing. Specifically, I think either a chi-squared test if we have really small numbers, like if we only do 5 trials total, a chi-squared test could tell us whether or not statistically the number of twitchers is related to the hydrocortisone amounts. Actually, the chi-squared would be for bigger counts. A Fisher's exact test, I haven't done one of those in a while because I always have higher counts. But if we had really small counts of worms, like if you plated for 50 but only like 10 of them survived, well, we can still use something to try to figure out the statistics. I haven't done one of those because I'm awesome and have high numbers all the time, but we'll see when we need. So 40 minutes at 11:15 at night for me. This is your project. Please let me know if you have questions along the way. A lot of culturing, a lot of chemistry. Not bad chemistry, not bad culturing. A lot of really advanced RNA interference and such. I will give you this slideshow so you can take notes on as much you like. I will also give you this video, whatever. I'm tired at this point. Let me know if you have questions.